The first topic we're going to cover is our genome. The human genome has many properties that make it distinct from other species' genomes, and here I'm going to cover a select few that the scientific community currently know about. But let's frame them as questions. How many base pairs is our genome? How many genes do we have? How big is a given gene? And what percentage of our genome is actually genes? So going to the first question, how many base pairs do we have? It's roughly 3.4 billion nucleotides. So within each cell we have about 3.4 gigabases. It's the equivalent of about 6 feet of DNA in each of our nuclei. How many genes do we have? Is it thousands, millions, hundreds of thousands? It's actually about 20,000. This was surprising when we first discovered this, as other arguably simpler species have even more genes than us. And so before we sequenced our human genome, we actually expected us to have many, many genes. And that's depicted here. The Human Genome Project was a government-funded project to sequence the human genome for the first time. And so before its launch, we had an estimate of about 100,000 genes. And the more we sequenced, we discovered that the number of genes we have is actually closer to 20,000. And so the question becomes, if we have so few genes, what is it that allows us to have such high levels of complexity compared to corn, for example? And one answer is multiple splicing. This is the phenomenon whereby one gene is post-transcriptionally modified in different ways and can create multiple proteins. So even though we have fewer genes, each of those genes can actually make different protein products. So, how big is a given gene? On average, genes in the human genome fall between 10 and 15,000 kilobase pairs, but there's high variation on this. The smallest genes can be less than 1,000 base pairs and can go up, the largest ones can go up to tens of thousands base pairs. What percentage of our genome codes for genes? It's extremely minimal. This was also surprising to scientists when it was first discovered, and what they hypothesized was that the remaining 99% was referred to as junk DNA, or non-functional. Didn't really have a purpose, it was just there. There was a theory that junk DNA existed as a form of redundancy to protect the coding in important parts of the genome. For example, if junk DNA is mutated, there's no actual consequences for the organism. However, we're finding more and more that these non-coding regions do actually have function for expression and regulation of genes. Areas like promoters, enhancers, and insulators are found in the non-coding regions and are vital for correct gene expression and cell function. Human genome has been sequenced. Who is it? We've talked a lot about these questions. They've been answered by us sequencing the genome for the first time. But human genome being sequenced, is it one person? Is it multiple people? And here's where we're going to delve a bit into the history. It's important to understand the historical context of some of these discoveries. So for example, we've talked about the Human Genome Project. Celera Genomics is a privately owned company that was also competing to sequence the human genome for the first time at the same time. The Human Genome Project and Celera Genomics were racing right in the late 1990s and early 2000s to be the first to publish. And they approached it with different methods. The Human Genome Project used many subjects, but Celera Genomics, the CEO, Craig Venter, decided that he would sequence his own genome. And so especially in the early 2000s, most of the genetic information we had on humans belonged to a single individual, Craig Venter. And although there were a few subjects from the Human Genome Project, there was obviously a problem with representation. Genetic information is obviously going to be different between males and females, different races, etc. And so since then, there have been many more humans sequenced to increase representation. Just a quick note. If you're interested in understanding the history of how our current knowledge of genetics is formed, There's a really good book called The Gene, An Intimate History. I highly recommend it. It's important not only to understand what technologies that we have now and how to use them, but how they develop. Because if you understand where a technology or where an institution comes from, you can potentially understand its limitations, its biases, and improve it in future. So in case you're interested, highly recommend. This kind of wraps up our section on the human genome. Keep in mind that we don't necessarily need you to memorize Celera Genomics or Craig Venter, but do want you to understand basic concepts. What percentage of our genome codes for genes, the fact that non-coding doesn't mean junk DNA, and in general the fact that humans have relatively few genes, but that doesn't mean that we're not complex.